Hi all, it's time for a quickie concept video. One of the things that's become very apparent and important over the years is to find what's called the different partners that proteins interact with. So different portions of a protein that may have an enzymatic activity will naturally bind with other proteins that have their own associated specific activities. And in turn, what we see are modifications of those two proteins that can activate them typically when they form these hybrid associations. So, what's the real significance of that? Well, we can look at it from the archaic, static perspective, or we can try to transform it into a dynamic perspective. So, this protein protein association, what it's going to do, it's going to physically localize protein interactions. It may be by a protein going into the nucleus and interacting with things there, as opposed to interacting with things in the cytoplasm. And these kinds of variations have profound effects in terms of being correlated with the cancer state, for example. So, how do we look at that with a slightly different perspective? Not negating that at all, but enhancing it through a dynamic perspective. Well, once you've localized two proteins together or one protein in a particular environment, what you're doing is now creating kind of a birthplace for a localization of a flow-dependent structure. This is a very, very interesting concept because what we're seeing then is different areas of the cells coming and going as nucleating agents for flow-dependent structures that are interacting with all the other flow-dependent structures to create a particular cell or collections of cells for tissue or collections of tissues for humans or collections of humans for society, etc. So anyway, it's a very interesting concept. Protein-protein interactions seed the location of dissapative structures. Bye.